Welcome back, everyone. Excited to dive into a new topic today, the world of genetics. And our guide for this deep dive is Genetics for Dummies. We'll be exploring the basics of heredity, how DNA is structured, and all that good stuff.
Sounds fascinating. Genetics really is the study of how traits get passed down through generations, right? It delves into that DNA code, which essentially serves as the blueprint for every living organism.
It really is amazing to think that every single living thing, from the tiniest bacteria to, I don't know, a giant whale, all share this same DNA code.
Yeah, it really speaks to the interconnectedness of everything on Earth. And what's even more incredible is just how far genetics has come. We're unlocking the secrets of that code, and it has huge implications for so many fields like healthcare, agriculture, even forensics.
Absolutely. So let's start with the foundation. Cells. Genetics for Dummies mentions two main types of organisms, prokaryotes and eukaryotes. And the big difference seems to be whether or not they have a nucleus. Can you break that down a bit more?
Sure. So prokaryotes, like bacteria, don't have a nucleus. Their DNA just kind of hangs out in a region called the nucleus. It's not enclosed. But eukaryotes, which include plants, animals, and fungi, they have a well-defined nucleus that keeps their DNA safe and organized.
And within those cells, Genetics for Dummies talks about these things called chromosomes. They're described as structures that hold DNA. What can you tell us about their role and how they're structured?
So think of chromosomes like threads made of DNA and proteins. They're super important because they make sure genetic information is packaged and distributed neatly during cell division. Each chromosome has a centromere, a kind of pinched point, that helps it attach to structures during division. And the ends are capped with telomeres, which are like protective caps made of repetitive DNA sequences. And, fun fact, different species have different numbers of chromosomes. Humans have 46, while fruit flies only have 8.
That's a big difference. The book also has this interesting experiment you can do at home using a strawberry to extract DNA. It shows how DNA is something tangible, not just this abstract idea. Can you walk us through how it works?
Yeah, it's a really cool visual. Basically, you crush the strawberry to break down those cell walls. Then you add soap and salt, which helps to burst the cells and release the DNA. The final step is adding cold alcohol, and this makes the DNA clump together and become visible as this white, stringy stuff.
Okay, so we've talked about where DNA is found, but let's zoom in on the DNA molecule itself. Genetics for Dummies describes it as a double helix, this twisted ladder shape. Can you explain more about the structure and those base pairs that form the rungs of the ladder?
The double helix is this incredible bit of molecular architecture. It's got two strands of nucleotides, the building blocks of DNA, all twisted around each other. And each nucleotide has a sugar molecule, a phosphate group, and a nitrogenous base. Now, there are four types of these bases: adenine A, thymine T, guanine G, and cytosine C. The key here is they pair up in a very specific way. A always pairs with T, and G always with C. This pairing is essential for keeping the genetic code intact when DNA replicates.
And this structure, this double helix, wasn't always known, right? The book mentions Rosalind Franklin, but points out that Watson and Crick often get most of the credit.
True. Rosalind Franklin's work using X-ray diffraction was absolutely critical. Her images were what revealed that helical structure. Unfortunately, her contributions weren't fully recognized while she was alive. There's been a lot of discussion about how credit is given, and frankly, all scientists involved in such discoveries deserve recognition.
Now, how does this genetic information actually get put to work? Genetics for Dummies talks about replication, transcription, and translation as this central dogma of molecular biology. Can you break down what that means?
The central dogma basically describes how genetic information flows within a cell. It goes from DNA to RNA, and then from RNA to protein. This flow is the foundation for all the processes that keep us alive.
Okay, so let's start with replication. The book mentions semi-conservative replication. What does that involve?
Well, it's the way DNA copies itself. The original DNA molecule unwinds, separating into two strands. Each strand then acts like a template to create a new matching strand. The end result is two identical DNA molecules, each with one original and one new strand. This ensures genetic information is passed accurately from one generation of cells to the next.
So each DNA molecule passes down half of its information to the next generation. That's incredibly efficient.
Exactly. And the accuracy of this process is super important. Errors during replication can lead to those mutations we talked about earlier, and they can have a range of effects. Some are harmless, but others can be really detrimental.
Genetics for Dummies also mentions telomeres. These are at the ends of chromosomes, and they get shorter each time DNA replicates. What implications does this have?
Telomeres act like those protective caps we talked about, preventing crucial genetic information from being lost during replication. But the problem is they get shorter with each cell division. And when they get too short, the cell can't divide anymore, it might stop functioning or even self-destruct, which is called apoptosis. This shortening of telomeres is actually one of the factors thought to contribute to aging.
That's fascinating. So there's a limit to how many times a cell can divide before those telomeres become too short.
Yeah, for most cells there is, but there are exceptions. Certain cells, like stem cells and germ cells, have an enzyme called telomerase. This enzyme actually lengthens telomeres, which allows these cells to keep dividing indefinitely.
That's incredible. So telomerase essentially makes those cells immortal. Okay, moving on to transcription. This is where we shift from DNA to RNA, specifically messenger RNA or mRNA. What's the role of mRNA in this flow of information?
Transcription is all about copying a specific segment of DNA, a gene, into mRNA. This mRNA molecule then carries those genetic instructions out of the nucleus, where the DNA is, to the ribosomes, which are in the cytoplasm. That's where protein synthesis happens.
It's like making a copy of a recipe from a cookbook. You don't copy the whole book, just the specific recipe you need.
Perfect analogy. During transcription, only the relevant part of the DNA is copied into mRNA, making sure the right instructions get to the ribosomes.
And just like a recipe has a start and stop point, genes have promoters and terminators, right?
Yes, absolutely. Promoters are sequences in the DNA that signal where to begin transcription, and terminators signal where to stop. This precise control is vital for making sure the cell functions correctly.
Now, the mRNA molecule isn't always a direct copy of the DNA, right? Genetics for Dummies mentions introns and exons, which make things a bit more complicated.
Introns and exons are actually pretty fascinating. Think of exons as the coding regions of a gene. They contain the information needed to build a protein. Introns, on the other hand, are these non-coding regions interspersed between the exons. They don't directly contribute to the protein's code.
So it's like having a recipe with extra information, almost like side notes, mixed in with the actual instructions. What happens to those introns?
Well, during mRNA processing, those introns are removed. It's called splicing. The exons are then joined together to create a mature mRNA molecule that's ready for translation. This splicing ensures that only the essential coding sequences are used to make the protein.
So why are introns even there in the first place if they're just going to be removed? What purpose do they serve?
That's a great question, and one that's puzzled scientists for a long time. There are several theories. One suggests they might play a role in regulating gene expression. Another proposes that they could have helped new genes evolve by allowing for the shuffling and recombination of those exon coding regions.
So even though introns don't code for proteins, they may have crucial roles in gene regulation and evolution. Genetics for Dummies also mentions alternative splicing. It sounds like cells have some flexibility in how they process that mRNA.
Alternative splicing is super interesting. It basically allows a single gene to produce multiple different proteins. During splicing, different combinations of exons can be included in the mature mRNA. This means that the same gene can give rise to various protein isoforms, each with potentially different functions.
So it's like having a base recipe, but you can modify it to create different variations of the dish. It's incredibly efficient.
Exactly. Alternative splicing massively increases the variety of proteins that can be made from a limited number of genes, and that's a big contributor to the complexity of cellular functions.
Okay, so finally we get to translation. This is where the mRNA message gets decoded to actually create a protein. It's like the final step in that central dogma. Can you walk us through this intricate process?
Translation happens at the ribosomes, which are basically complex molecular machines for protein synthesis. The mRNA molecule binds to the ribosome, and its sequence of codons, which are 3 nucleotide units that specify which amino acid to add, is read. Transfer RNA molecules, or tRNAs, come into play here. Each tRNA carries a specific amino acid, and they bind to the mRNA codons based on those base pairing rules we talked about. The ribosome then helps link the amino acids together in the order specified by the mRNA.
So it's like an elaborate assembly line. The mRNA is the blueprint, and the tRNA molecules are delivery trucks, bringing in the amino acid building blocks in the right order.
I like that. The ribosome moves along the mRNA, codon by codon, and the tRNAs deliver the matching amino acids, adding them to the growing protein chain. This continues until a stop codon is reached, signaling the end of protein synthesis.
It's amazing how this whole chain of events, from replication to transcription to translation, allows the information in DNA to be expressed as functional proteins that do all the work in our cells.
These processes are fundamental to all life forms. They drive everything that happens inside cells, and they ensure the continuity of life from generation to generation.
Now, to switch gears a bit, let's talk about Gregor Mendel, who's often called the father of genetics. His work with pea plants really laid the groundwork for understanding how traits are inherited.
Mendel was a pioneer. He was incredibly meticulous in his experiments. He crossed pea plants with different traits and carefully tracked how those traits were passed down over many generations. His observations led him to deduce three fundamental laws of inheritance: the law of segregation, the law of independent assortment, and the law of dominance.
Those laws are considered cornerstones of genetics. Can you explain each one in more detail?
Of course. The law of segregation states that each individual has two alleles, or versions, of a gene for each trait. During the process of creating gametes, like sperm and egg cells, these alleles are separated, so each gamete only gets one allele for each trait.
So it's like each parent shuffles their genetic deck and deals out half of their cards to their offspring.
Exactly. This random segregation of alleles ensures that offspring inherit a mix of traits from both parents, creating genetic diversity.
The law of independent assortment sounds a bit chaotic, but it basically tells us how multiple traits are inherited, right?
Yes, it states that the inheritance of one trait doesn't affect the inheritance of another. So the alleles for different traits are sorted into gametes independently of each other.
So whether you inherit your mom's brown eyes doesn't determine if you'll also get her curly hair. Those genes are sorted independently.
Precisely. This independent assortment further amplifies the diversity of trait combinations in offspring, making each individual genetically unique.
Finally, we have the law of dominance. This explains why some traits seem to win out over others. What's the mechanism behind that?
The law of dominance says that some alleles are dominant over others. This means that a dominant allele will be expressed even if an individual only has one copy of it. The other allele, the recessive one, will only be expressed if the individual inherits two copies.
So if you inherit one allele for brown eyes, which is dominant, and one for blue eyes, which is recessive, you'll have brown eyes. The brown allele masks the blue one.
That's right. The dominant allele exerts its influence even when the recessive allele is present. But it's important to note that the recessive allele isn't gone. It can still be passed down to future generations.
It's like a hidden genetic potential that could reemerge under the right circumstances.
Exactly. So these three laws, segregation, independent assortment, and dominance, they give us a basic framework for understanding how traits are passed down.
But as we delve deeper into genetics, things aren't always as clear-cut as Mendel's laws might suggest, right? Genetics for Dummies talks about some exceptions, like incomplete dominance and codominance.
You're absolutely right. Incomplete dominance happens when neither allele is completely dominant. The resulting phenotype is a blend of the two. A classic example is flower color. If you cross a red flower with a white flower and the offspring are pink, that's incomplete dominance. Neither the red nor the white allele is fully expressed, you get a mix.
So it's like mixing paint, red and white make pink.
How is codominance different?
In codominance, both alleles are fully expressed. Think about the ABO blood group system. Someone can inherit both the A and B alleles, and both are expressed, resulting in blood type AB. Neither allele masks the other. They both contribute equally to the phenotype.
It's like both alleles are equally loud, each contributing their distinct characteristics to the phenotype. The book also talks about multiple alleles, which seems to make things even more complex. Multiple alleles mean there are more than just two possible alleles for a gene in a population. The ABO blood group system is a great example of this. You have the A, B, and O alleles, and this leads to a wider range of potential genotypes and phenotypes.
So instead of just two options, there's a whole pool of possibilities. And then there's epistasis, which sounds like a complex interaction between genes.
Epistasis is when one gene influences the expression of another gene. It's like one gene is acting as a supervisor, controlling whether or not another gene gets to do its job.
The book uses coat color in horses as an example. One gene determines if there's any pigment at all, while others control the specific color. It really shows that genes don't always act alone. Their expression can be interconnected in intricate ways.
Exactly. And all these complexities, incomplete dominance, codominance, multiple alleles, epistasis, they highlight how intricate these interactions are. They all contribute to the incredible diversity of traits we see in living organisms.
So while Mendel's laws give us a basic understanding, they're just the beginning when it comes to the complexities of genetics.
That's a great way to put it. As we continue to unravel the mysteries of genetics, we gain a deeper appreciation for the amazing complexity of life and the mechanisms that drive its diversity.
That's a great point. Mendel's work was foundational, but there's so much more to uncover. Genetics for Dummies then gets into mutations. They often get a bad rap. But what exactly are mutations and what role do they play?
Mutations are basically permanent changes in that DNA sequence. And while some mutations can be harmful, they're actually the main source of genetic variation. Without mutations, there wouldn't be the diversity of life we see.
So mutations are actually crucial for evolution. The book emphasizes how mutations provide that raw material for natural selection to work with, leading to the amazing diversity in nature.
Exactly. There are different types of mutations. Point mutations are small-scale changes affecting just one base pair in the DNA. Like a typo in a sentence. It could be a substitution, swapping one base for another, an insertion, adding an extra base, or a deletion, removing a base. And those little changes can have big effects on the resulting protein.
And then there are chromosomal mutations, which sound much more significant. What happens there?
Chromosomal mutations involve bigger changes. We're talking about whole chunks of chromosomes being affected. These can be translocations where a piece of one chromosome breaks off and attaches to another, inversions where a segment flips, duplications where a segment repeats, and deletions where a segment gets lost.
I can see how chromosomal mutations would likely have bigger consequences than point mutations.
Usually, yes. Chromosomal mutations can disrupt many genes at once. That can dramatically change how a cell functions. A lot of genetic disorders actually come from these kinds of mutations.
The book uses cystic fibrosis and sickle cell anemia as examples, showing how impactful mutations can be.
Cystic fibrosis, for example, happens because of mutations in a gene that controls salt and water transport in cells. This causes a buildup of thick mucus in the lungs and other organs, which makes it hard for them to work properly. Sickle cell anemia, on the other hand, is due to a mutation in the gene for hemoglobin. That's the protein that carries oxygen in our red blood cells. This mutation causes the red blood cells to become sickle-shaped, which hinders their ability to carry oxygen effectively.
It's clear those conditions have a significant impact on someone's health. Are mutations predetermined? Or do things like environmental factors play a role?
Well, some mutations are spontaneous, happening naturally during DNA replication. It's a remarkably accurate process, but not perfect. And then there are induced mutations caused by exposure to things like radiation, certain chemicals, or even some viruses.
So our cells are under constant attack, both from within and from the outside world. It's remarkable they're able to maintain their integrity.
Cells have really sophisticated repair mechanisms. They're constantly working to fix errors in DNA and protect that genetic code.
Like having a dedicated team of proofreaders and editors constantly scrutinizing the DNA for errors.
That's a good way to think about it. This balance between introducing variations through mutations and keeping things stable through DNA repair is crucial for survival and evolution.
Shifting gears to a more somber note, the book points out that while mutations are the source of genetic variation, they can also lead to genetic disorders.
Right. Genetic disorders are conditions caused by abnormalities in our DNA. It could be anything from single gene mutations, like in cystic fibrosis or sickle cell anemia, to chromosomal problems where whole chromosomes or pieces of them are missing, duplicated, or rearranged.
Down syndrome, for instance, is caused by an extra copy of chromosome 21. That shows the profound impact such abnormalities can have.
Absolutely. The effects of a genetic disorder really depend on the specific gene or chromosome involved and the nature of the abnormality. Some disorders might have mild symptoms that are treatable, others are severe and debilitating.
The book talks about the challenges of diagnosing and treating genetic disorders and mentions genetic testing and counseling. Can you elaborate on those?
Genetic testing is how we analyze someone's DNA to find abnormalities linked to a specific disorder. You can test different samples like blood, saliva, even tissue. Genetic counseling is more about helping people and families understand what genetic disorders mean for them.
So testing gives you the data, counseling helps you make sense of it.
Precisely. Counselors help people understand their risks, explain test results, and make informed decisions about family planning and healthcare.
It's amazing how genetic information is being used to personalize medical treatment now.
It is. Pharmacogenomics, for example, looks at how your genetic makeup influences how you respond to medications. By understanding these genetic variations, doctors can personalize drug prescriptions and dosages, making treatments more effective and reducing the risk of bad side effects.
Genetics for Dummies highlights that adverse drug reactions are a serious problem, even leading to hospitalizations and deaths. Tailoring treatments based on genetics could really improve patient safety.
Absolutely. Pharmacogenomics holds a lot of promise. Identifying who will benefit most from a drug or who's at risk of side effects can greatly improve treatment plans and patient outcomes.
Now, zooming out to look at populations, Genetics for Dummies also gets into population genetics. Can you tell us about this area of study?
Population genetics is all about the genetic variation within and between groups of individuals. It tries to understand where this variation comes from, how it's maintained, and how it changes over time.
The book mentions allele and genotype frequencies, which sound pretty technical. Can you clarify what they are?
Sure, allele frequency is just how often a specific allele, a version of a gene, shows up in a population. Say an allele appears in 50% of individuals, its frequency is 0.5 or 50%. Genotype frequency tells you how often a particular combination of alleles, a genotype, appears. So if 25% of individuals have two copies of a certain recessive allele, that genotype's frequency is 0.25 or 25%.
So allele frequencies tell us about individual gene versions, and genotype frequencies show us how those versions combine within a population.
Exactly. These frequencies are important for understanding a population's genetic diversity and how it changes.
The book introduces this concept called the Hardy-Weinberg law. Sounds important in population genetics.
The Hardy-Weinberg law describes a theoretical scenario of genetic equilibrium. It basically says that in a large, randomly mating population, if there aren't any evolutionary forces acting on it, allele and genotype frequencies stay the same across generations.
So it's a baseline, right? If you see differences from Hardy-Weinberg in a real population, it means something's going on, some kind of evolutionary pressure is at play.
Precisely. Deviations mean factors like mutation, gene flow, genetic drift, or natural selection are influencing allele and genotype frequencies.
That's a lot of factors. Can you give us a quick overview of each one?
Of course, mutations, we've talked about, introduce new alleles. Gene flow is the movement of alleles between populations, usually through migration. Genetic drift is random changes in allele frequencies, which is more important in small populations. And natural selection favors the survival and reproduction of organisms with advantageous traits.
Like sculptors shaping the genetic landscape of populations.
A perfect analogy. By studying these forces, we understand how populations evolve and adapt.
The book emphasizes the importance of genetic diversity for a healthy population. Why is diversity so vital?
Think of genetic diversity as an insurance policy. A population with high diversity is more resilient. They can better handle changes like new diseases or shifts in the climate because there's a greater chance someone has a trait that's beneficial in the new situation.
So a diverse gene pool helps a population survive tough times.
Exactly. Conversely, populations with low diversity are much more vulnerable. They lack the genetic flexibility to adapt to new threats and are more susceptible to extinction.
The book also touches on conservation genetics. How does genetics help in protecting endangered species?
Conservation geneticists use genetic analysis to understand the threats facing endangered populations and develop conservation strategies. They'll analyze genetic data to estimate population size, see how genes are flowing between isolated groups, and check the level of inbreeding, which can be a problem for small populations.
So genetics gives them insight into the past, present, and future of these populations.
Precisely. If they find a population with low diversity or facing genetic threats, they can design strategies like habitat restoration, captive breeding, or even translocations to bring in new genes and improve the population's health.
It's amazing how genetic tools are being used in conservation efforts. Turning to a different area, Genetics for Dummies discusses forensic genetics and the use of DNA in solving crimes.
Forensic genetics is fascinating. It uses genetic analysis in legal situations, relying on the fact that everyone, except identical twins, has a unique DNA profile.
The book describes DNA fingerprinting, which creates a unique genetic fingerprint for an individual. How does that work?
DNA fingerprinting focuses on these short tandem repeats, or STRs. They're short DNA sequences repeated in a row at specific locations in the genome, and the number of repeats varies greatly from person to person.
So by looking at enough STRs, you get a profile that's highly likely to be unique, just like a regular fingerprint.
Exactly. The chance of two unrelated people having the same STR profile is incredibly small.
Genetics for Dummies mentions CODIS, the Combined DNA Index System. What's the purpose of that database?
CODIS is a powerful tool for law enforcement. It lets them compare DNA profiles from crime scenes to a database of profiles from known individuals, like convicts or arrestees.
So a match could link a suspect to a crime or clear an innocent person.
Exactly. CODIS has revolutionized how we investigate crimes, providing solid evidence that can really make a difference in the pursuit of justice.
DNA evidence has changed everything in forensics. Genetics for Dummies also touches on genetic engineering. It's a field that's captured our imaginations and raised some concerns.
Genetic engineering is about manipulating an organism's genes, introducing new traits or altering existing ones. It has huge potential in medicine, agriculture, and biotechnology.
The book focuses on transgenic organisms. Those are organisms that have had foreign DNA inserted into their genomes. How's that done?
We use recombinant DNA technology. It's like cutting and pasting DNA. Scientists can create new combinations of genetic material from different sources and then insert these recombinant DNA molecules into an organism. Often they use vectors like modified viruses or bacteria to carry the new DNA.
The book gives the example of using a bacterium called Agrobacterium tumefaciens to create transgenic plants.
That's right. Agrobacterium naturally inserts its DNA into plant cells. Scientists figured out how to use that natural mechanism to deliver desirable genes into plants, creating crops with improved traits.
Like making crops resistant to herbicides, insects, or diseases. That could really change agriculture.
The potential is enormous. Transgenic crops could boost yields, reduce pesticide use, and make food more nutritious. But it's important to acknowledge the public debate around genetically modified foods and the concerns about safety and ecological impact. Genetics for Dummies covers that too.
Open discussion about these issues is critical, especially as genetic engineering technology continues to advance. The book also talks about transgenic animals.
Transgenic animals are created similarly to plants by inserting foreign DNA into fertilized eggs. They're incredibly useful in research, drug production, and even agriculture. For research, especially with mice, they're used to model human diseases, test new drugs, and figure out what different genes do.
The book mentions transgenic mice as models for human diseases like Alzheimer's and cancer.
Exactly. These models are essential for understanding diseases and developing treatments. Transgenic animals like goats can even be used to produce therapeutic proteins in their milk.
The book uses the example of producing antithrombin, a blood-clotting protein, in goat's milk.
It's a really efficient and cost-effective way to produce large amounts of complex proteins. And in agriculture, transgenic animals are being developed with traits like enhanced muscle mass, disease resistance, or better milk production. However, just like with GM crops, there are ethical considerations around the use of transgenic animals.
Animal welfare needs to be a top priority, and ethical guidelines should be strictly followed. Now, moving to a topic that often sparks a lot of discussion, the book talks about cloning.
Cloning is creating genetically identical copies. It can be genes, cells, even whole organisms, and there are different types of cloning, each with its own set of uses and ethical implications.
Genetics for Dummies focuses on reproductive cloning, where you aim to create a genetically identical copy of an animal.
Yes, Dolly the sheep in 1996 is the famous example. She was the first mammal clone from an adult somatic cell, which is any cell in the body other than sperm or egg cells. That proved it was possible to create a genetic copy of an animal using a body cell.
The book describes the process of somatic cell nuclear transfer. How does that work?
Somatic cell nuclear transfer, or SCNT, is where you take the nucleus out of an egg cell and replace it with the nucleus from a body cell of the animal you want to clone. You then stimulate that egg to divide and develop into an embryo, which is implanted into a surrogate mother. If all goes well, the surrogate gives birth to a clone of the animal that donated the nucleus.
It's a complicated process with lots of ethical questions, especially the possibility of cloning humans.
Definitely. Human cloning raises massive ethical dilemmas about identity, individuality, and the potential for misuse. It's something we need to have a serious societal discussion about, as well as about other reproductive technologies. Genetics for Dummies even touches on epigenetics.
Epigenetics. Can you remind us about that again?
Epigenetics is how environmental factors can influence gene expression without changing the actual DNA sequence. It's like adding a layer of control on top of our genes.
So our environment and experiences can have a lasting impact on how our genes work.
Exactly. Things like diet, stress, toxins, even social interactions can influence epigenetic changes, impacting gene expression, and potentially contributing to diseases like cancer, heart disease, or mental illnesses.
The book mentions the developmental origins of health and disease concept, that our early life experiences, even in the womb, can affect our health later.
Right. It suggests that what happens during key developmental stages, especially prenatally and in early childhood, can alter epigenetic programming and influence a person's susceptibility to chronic diseases later in life.
So our health is influenced by a combination of our genes and our environment throughout life.
Exactly. Epigenetics has changed how we think about nature and nurture, highlighting how interconnected our genes, environment, and health really are.
It really is amazing how much we've learned about genetics, starting with Mendel's work, and now we're mapping entire genomes. Genetics for Dummies also talks about what's hot in genetics today, and it's mind-blowing. We touched on pharmacogenomics earlier. Can you unpack how our genes affect how we respond to medications?
Pharmacogenomics is all about that connection between our genes and how we react to drugs. It's the idea that variations in our genes can change how our bodies process and use those medications. These differences can make a drug more or less effective, change the likelihood of side effects, even impact the ideal dose for a person.
So, if doctors understand a patient's genes, they can personalize prescriptions, getting the best results and minimizing risks. That's huge.
Exactly. Some genes, for example, make enzymes that break down drugs. Variations in those genes mean some people metabolize a drug quickly, making it useless, while others metabolize it slowly, leading to a buildup and potential side effects.
The book also talks about gene therapy, which seems like science fiction. It's about actually fixing genetic defects to cure diseases.
Gene therapy is a rapidly developing field with huge potential to change how we treat genetic disorders. It involves introducing, replacing, or even turning off genes to treat or prevent disease. There are different methods, but the goal is to get those therapeutic genes into the patient's cells.
So, like
Yeah.
fixing a typo in the DNA code itself.
A good way to think about it. A common approach uses viral vectors. These are modified viruses that carry the good gene into the target cells. There are also non-viral methods using things like nanoparticles or liposomes to deliver the gene.
The book mentions gene therapy being researched for cystic fibrosis, hemophilia, and even some cancers. It's incredible to think we might actually cure these diseases by fixing the genes.
There are challenges for sure, like ensuring the gene delivery is safe, that it's effective, and making sure the gene gets to the right cells. But gene therapy has the potential to change everything. It's a shift from just treating symptoms to addressing the root cause of a disease.
Another hot topic the book covers is the human microbiome, all those microbes living in and on our bodies.
The human microbiome is this amazing, complex ecosystem of trillions of bacteria, viruses, fungi, the whole gang living in and on us. And it plays a massive role in our health. It affects everything from digestion and nutrient absorption to our immune system and defense against pathogens.
We're like walking ecosystems with more microbial cells than human cells. That's crazy. What have we learned about this microbiome?
Well, we know it's incredibly diverse, and the composition varies from person to person. Diet, lifestyle, genetics, environment all play a role. And we're finding that changes in the microbiome are linked to all sorts of health problems, from obesity and diabetes to gut issues, allergies, even some neurological conditions.
The book even mentions fecal transplants as a treatment for C. difficile infections. That's wild.
Fecal microbiota transplantation, or FMT, is actually very effective for recurrent C. difficile infections. It works by transferring fecal matter from a healthy donor to someone with a disrupted microbiome. Success rates can be over 90%, which shows how important a healthy microbiome is.
It's a good reminder that our health is tied to the health of all those microbes living inside us. We're not just individuals, we're walking communities.
Exactly. We need to consider the interaction between our genes, microbiome, and environment to really advance healthcare and find more personalized ways to prevent and treat diseases.
Genetics for Dummies also brings up the issue of antibiotic resistance. It's a bit scary to think about the evolution of superbugs.
Antibiotic resistance is a serious threat, and it's driven by the overuse and misuse of these drugs. Bacteria are constantly evolving and adapting to survive.
The book describes how bacteria can get resistance genes, either through mutations or from other bacteria, through this process called horizontal gene transfer.
That's right, mutations happen naturally, and some can make bacteria resistant to an antibiotic. Those resistant bacteria survive, multiply, and pass on those genes. That's how you get antibiotic-resistant strains.
So the more we use antibiotics, the more we encourage the spread of those resistant bacteria.
Exactly. Bacteria can also get resistance genes directly from other bacteria. This is called horizontal gene transfer. They swap genetic material, including resistance genes, through different mechanisms. It's like they're sharing weapons to fight off our antibiotics.
So it's a constant arms race. As resistance spreads, infections become harder to treat, leading to longer hospital stays, higher healthcare costs, even death.
It's a major public health concern. We need to be more careful about antibiotic use, only using them when necessary. At the same time, we need to develop new antibiotics and alternative therapies to fight those resistant bacteria.
We talked about epigenetics before, but it's worth revisiting how our environment can affect our genes.
It really is fascinating. Epigenetics is about those modifications that control gene expression without changing the DNA sequence itself. It's like an extra layer of regulation deciding which genes are on or off. And those changes, things like DNA, methylation, or histone modification, are dynamic and respond to what's happening around us.
So what we do and experience can actually change how our genes work.
Exactly. Studies have shown that diet, stress, toxins, even our social lives can influence those epigenetic modifications, impacting gene expression and potentially increasing the risk of diseases like cancer, heart disease, and mental illness.
Genetics for Dummies mentions the idea that early life experiences, even before birth, can have lasting effects on our health.
It's called the developmental origins of health and disease hypothesis. It proposes that environmental factors during those crucial periods of development, especially in the womb and early childhood, can set the stage for health problems later in life. It's all about those epigenetic changes.
So our health is a combination of our genes and all the things we're exposed to throughout life.
That's the key takeaway. Epigenetics shows us how nature and nurture work together to shape who we are. Our genes are important, but they're not the whole story.
This deep dive into genetics, guided by Genetics for Dummies, has been incredible. It's clear that genetics is a vast and complex field touching everything from our health to the way we solve crimes. And it's constantly changing, pushing the limits of scientific understanding and bringing up important ethical questions.
I absolutely agree. Genetics has opened up a whole new level of understanding about ourselves and the world around us. It's full of potential, from transforming healthcare to revolutionizing agriculture, and it makes you appreciate the wonders and complexity of life.
Thank you for joining us on this journey through the world of genetics. We hope you've gained a new appreciation for this amazing science.